Hello everyone, our cancer study presentation is on osteosarcoma. My group mates are Luigi, Gutierrez, Daisy Andrade, and myself, Brianna, Rangel. I'm going to start off with the disease summary and cell tissue of origin. Osteosarcoma is a malignant bone tumor that comes from cells in the bone-forming osteogenic lineage. It is the most common primary bone cancer and usually develops around bones like femur, tibia, and humerus, especially near the metaphyseal growth place where bone growth is most active. At the cellular level, osteosarcoma is thought to arise from mesenchymal stem cells or their early osteogenic progenitors. Normal bone-forming cells that pick up cancer-promoting mutations in key tumor suppressor genes like TP53 and RB. These changes can cause the cells to lose normal growth control and start producing abnormal bone osteoid, which is the hallmark of the disease. Within the tumor, a smaller group of cells called cancer stem cells, CSCs, can self-renew, resist chemotherapy, and are believed to drive relapse and metastasis. These CSCs are strongly influenced by their bone microenvironment, which are growth factors, hypoxia, and signaling pathways like WNT, NOTCH, PI3K, and TGFB, which is why newer therapies are not only targeting the bulk tumor, but also these stem-like cells to improve outcomes for patients. Now, moving to the epidemiology, osteosarcoma is the most common primary bone cancer and arises from primitive bone-forming mesenchymal cells. Its epidemiology shows a distinct pattern with overall incidence rates of about 4.0 cases per million per year in children 0 to 14 years old and 5.0 per million in those 0 to 19 years old, although it accounts for only 2.4% of childhood cancers. It follows a predictable bimodal age distribution. The first and largest peak occurs in adolescents ages 10 to 14, closely aligning with the rapid bone growth of puberty, suggesting a strong connection between growth spurts and tumor development. A second smaller peak appears in adults over 65, often linked to underlying conditions such as Paget's disease. Incidence also varies by sex and race, occurring more frequently in males, 5.4 per million, than females, 4.0 per million, and more commonly in Black, 6.8 per million in Hispanic, 6.5 per million populations, than in Caucasian populations, 4.6 per million. Osteosarcoma remains a significant contributor to childhood cancer mortality, representing 8.9% of bone and joint cancer deaths. Despite its severity, death rates have been steadily declining by about 1.3% per year, and the current 5-year survival rate is approximately 68%, with outcomes strongly influenced by age, presence of metastasis, tumor location and size, chemotherapy response, and the ability to achieve complete surgical removal.
So now I'm going to be talking about cancer staging of osteosarcoma. For this slide, I'm going to clearly explain how osteosarcoma is staged and why each part of the staging system matters. Osteosarcoma uses the AJCC-TNM system, which is specifically made for bone tumors. The first part of the system is the T classification, which focuses on the size of the primary tumor. If the tumor is 8 centimeters or smaller, it is labeled as T1. If it's larger than 8 centimeters, it's T2. There's also a T3 category, and this is used when there are multiple separate tumor areas within the same bone, which is something unique to bone cancers and indicates a more complex tumor pattern. Next, we look at the N classification, which refers to lymph node involvement. Osteosarcoma almost never spreads to lymph nodes, so most cases are no. But if a lymph node involvement does happen, it's considered very advanced and is associated with the worst prognosis. The M classification describes whether the cancer has metastasized. Osteosarcoma has a strong tendency to spread to the lungs, which is labeled as M1A. If it spreads to other organs or additional bones, it's labeled as N1B. Lung metastasis is especially important because it significantly worsens outcomes. All of these factors come together to the overall form of stage. Stage 1 tumors are low grade and localized. Stage 2 tumors are high grade but still contained within the bone. And stage 3 means that there are multiple tumor spots in the same bone. Stage 4 indicates metastatic disease, usually to the lungs. Staging is incredibly important because it directly influences the treatment plan and survival expectations. According to the AJCC Cancer Staging Manual and data from the National Cancer Institute, localized osteosarcoma has a 5-year survival rate of about 60 to 70%, while metastatic osteosarcoma drops to around 20 to 30%. This is why staging plays such a major role in determining the intensity of chemotherapy, the type of surgery, and how aggressively the disease needs to be managed. Now for the next slide of slide 4, which is the cellular aspects of osteosarcoma, I'm going to be talking and breaking it down what is actually happening at the cellular level of osteosarcoma. Osteosarcoma develops from immature osteoblast lineage cells, which are supposed to form healthy bone. When these cells become cancerous, they lose their normal regulation and start producing malignant osteoid, which is a disorganized and abnormal form of bone matrix. This is considered one of the most defining features of osteosarcoma. A major reason for these cells lose control is because of the mutations in key tumor suppressor genes. One of the most important genes affected is P53, which normally acts as a quality control system. When cells have DNA damage, P53 either repairs the damage or signals the cell to undergo apoptosis. In osteosarcoma, P53 is often mutated or completely lost, which means damaged cells can continue dividing instead of being eliminated. Another crucial gene is the RB gene, which controls the G1 to S phase transition of the cell cycle. When RB is inactive, cells move through the cell cycle too quickly, causing uncontrolled proliferation. Research from Natural Reviews Cancer and other sources show that these genetic changes contribute to the high level of genomic instability seen in osteosarcoma. These tumor cells are also very aggressive in how they interact with the bone environment. They release matrix metalloproteinases, or MMPs, which break down the bone around them and make it easier for the tumor to expand. They also heavily rely on glycolysis, even when oxygen is present, because this metabolic shift allows them to generate energy quickly. Osteosarcoma tumors also stimulate new blood vessel growth, which provides nutrients that support rapid expansion. All of these cellular changes help explain why osteosarcoma so commonly spreads to the lungs. The tumor cells are highly invasive, capable of entering the bloodstream, and they tend to survive and grow once they reach the pulmonary environment.
I'll be talking about molecular aspects. First of all, osteosarcoma is a case of extreme genomic instability rather than it being a single consistent driver mutation. In many of these cases, one can see massive chromosomal upheaval with large-scale structural rearrangements, translocation, deletions, and amplifications. In some cases, events classified as chromotripsis, which is basically chromosome shattering and reassembly, or more complex chromoanagenesis has been documented. Among the most recurrent consequences of instability is the loss of function of major tumor suppressor pathways. One of the most important ones being TP53 gene, which is a master regulator of DNA damage response, apoptosis, and cell cycle arrest. This occurs through point mutations, but even more frequently through structural rearrangements or deletions. Another important gene is the RB1 gene, which encodes the retinoblastoma protein that controls the G1 to S cell cycle checkpoint. It's also frequently lost and inactivated. The loss of these guardrails basically removes critical checks on proliferation, apoptosis, and DNA integrity, basically allowing for accumulation of more mutations and aggressive tumor growth. Also, osteosarcoma cells frequently acquire oncogenic alterations and signal pathway changes. For example, activation of the PI3K-mTOR pathway, which is a key survival and growth pathway, has been identified as commonly being vulnerable across many OS tumors. Overexpression of c-Myc and dysregulation of other genes involved in proliferation, and differentiation and survival has also been an issue. Finally, there's also been a lot of evidence that's been accumulating over other genes and regulatory RNAs, which include various tumor suppressors or developmental regulators, some being Notch, WNT, or other pathways, as well as microRNAs such as MIR-21 or MIR-34A. These also alter in osteosarcomas. They may modulate invasiveness, resistance to apoptosis, metastatic capability, and chemoresistance by affecting signaling such as MAPK or PI3K-AKT. Next, I'll be talking about treatment and results. First of all, for standard therapy, osteosarcoma treatment is built on a multimodal approach, basically combining chemotherapy and surgery. The current standard of care is the MAP regimen, which includes high dose methotrexate, doxorubicin, and cisplatin. The protocol has been established since 1980s through clinical trials and still is foundational for therapy today. There's also neoadjuvant chemotherapy, basically meaning treatment given before surgery, which helps to shrink the tumor, eradicate micrometastatic diseases, and is critical predictor of the outcome. Tumors demonstrating less than 90% necrosis, basically poor histological response, after the neoadjuvant chemotherapy have significantly lower survival rates compared to good responders. There's also surgical management, which aims for wide local excisions with negative margins. Basically, it's trying to preserve the bone and only cutting out the affected area. And due to greater techniques and with the chemotherapy, it has become a lot easier to do that. Next, for alternative approaches for patients with relapsed or refractory diseases, outcomes remain poor. Second line therapies often include ifosfamide, eptoside, or high dose regimens, but durable responses are uncommon. For metastatic diseases, especially those in the lungs, there is a much worse prognosis. In the lungs, it can improve survival in select patients, but it's not likely. Because osteosarcoma is a genomically chaotic cancer with few treatable mutations, targeted therapies such as IGF-IR inhibitors or mTOR inhibitors have some modest benefits, but no actual dramatic breakthroughs. For immunotherapy, which include checkpoint inhibitors, there has been limited success so far because osteosarcomas tend to have a low mutational burden, but ongoing trials are testing combinations that may improve response. And basically, for treatment results, the localized cases, there's a 60 to 70% 5 year survival rate, which is really good. Or for metastatic osteosarcomas, especially in the lungs, it decreases to around 20 to 30% for a 5 year survival. So it's kind of a lot lower. And weak response to neoadjuvant chemo leads to worse prognosis. So, continued research into immunotherapy and molecular targeted strategies is essential to improve survival at this point. These are our sources, and thank you so much for your time.